Hi everyone. In this video, we're going to be explaining inorganic compounds. To get us started, we're going to define nutrients and metabolites. So nutrients are essential molecules or elements obtained from the diet. When we say the word essential, we mean that it has to be consumed because the body is not capable of synthesizing it and really can't survive without it. Metabolites are all of the molecules that can be synthesized or broken down inside of the body. Nutrients and metabolites can be classified as inorganic compounds and organic compounds. So inorganic compounds typically do not contain carbon and hydrogen together. You might see carbon or hydrogen, but you just won't see them in the same molecule. Common examples of inorganic compounds are carbon dioxide, oxygen, water, acids and bases, and salts. Organic compounds are primarily composed of carbon and hydrogen, and then a lot of times we'll see things like oxygen and nitrogen as well. But organic compounds, carbon is the major contributor, and there'll be lots of carbons that are strung together. So organic compounds tend to be larger and more complex than inorganic compounds. The examples include carbohydrates, lipids, proteins, nucleic acids, and the molecules that contain high energy bonds. High energy bonds are essential to energy transfer in the human body. So in this video, we're going to consider the inorganic compounds, and in the next video, we'll consider the organic compounds. So first, let's start by talking about carbon dioxide and oxygen. So oxygen is consumed by cells in the body and absorbed by the lungs. Carbon dioxide is produced by metabolic activity and released by the lungs. And both gases are transported in the blood. So we're going to see throughout our study of human physiology many instances where we have to talk about oxygen and carbon dioxide and the ability of these gases to arrive at the appropriate destinations in adequate concentrations in order to allow the body to continue to survive. The next inorganic compound we want to talk about is water. Water constitutes about 2/3 of total body weight. Water is a component in many chemical reactions occurring in the body. Water has a high heat capacity, so that means it has the ability to absorb and maintain heat and stabilize body temperature. And also, water is a solvent. It dissolves many molecules, creating solution, and it's a medium for many chemical reactions. So I want to take a few moments to expand on the idea of a solvent and talk a little bit about solutions. So we'll just draw a glass beaker, and in the glass beaker we'll have some water, and then we'll also have, let's just for the sake of the example, say that we're going to dissolve some salt particles in the water. So a solution is a uniform mixture of solute and solvent. So solute is basically the dispersed or dissolved particles. In this case, it's going to be the salt molecules. So it's actually going to be sodium ions and chloride ions, and we're going to get into that in a moment. And the solvent is the medium in which the atoms or ions or molecules are dissolved. When we say uniform mixture, what that means is that we could take a small sample of that water containing the solute, in this case the salt, and that sample would have the exact same concentration or ratio of salt to water as would the entire rest of the solution in the glass beaker. So it's very different from something like a suspension, which activated charcoal is a good example of a suspension, where you might have particles that don't really necessarily dissolve and maybe a little less uniform in their dispersion. Now, we mentioned previously that an ion is a charged particle, and when we talk about ions and ionic compounds, we also would like to talk about ionization. So ionization is equivalent to dissociation. It means that when we place an ionic compound in water, the ionic compound is going to separate, and the positive and negative charges associated with the ions will interact with the positive and negative ends of polar molecules. So we described initially something like salt. So we'll use salt as our example here. Sodium chloride. We describe that as an ionic bond such that the positive and the negative charges are attracted to each other like magnets. If we place a molecule of salt in water, the sodium ions and the chloride ions are going to separate from each other. So essentially what happens is the sodium suddenly becomes surrounded by water molecules, and the water molecules are going to orient themselves in such a way that the partial negative charge associated with the oxygen is going to face the positive charge associated with the sodium. So the water molecules essentially create a hydration sphere around that sodium ion, and its function is to keep that sodium ion dissolved. So now you can see that if we were to draw a chloride ion, it actually doesn't have good access to that sodium ion because the sodium ion is completely surrounded. Additionally, the chloride ion is going to be surrounded, but because the chloride ion is negatively charged, the water molecules will orient themselves so that the partial positive charges associated with the hydrogens will be facing that chloride. So not only can the chloride not access the sodium because the sodium is surrounded, but also the chloride possesses its own hydration sphere. With that said, this is an opportune time to introduce the word electrolyte. So an electrolyte is a soluble inorganic molecule whose ions will conduct electrical current in solution. So sodium chloride is an electrolyte, and when sodium ions and chloride ions are placed into solution, they have the ability to conduct electrical current. Electrical current allows for many body functions, things like neuromessaging, the transmission of action potentials, the heartbeat, muscle contraction. All of these things are permitted to function by flow of electrical current in the human body. So although it's a little bit premature because we haven't explained these concepts yet, it is a super critical point because it's something that really, really explains a lot of how the human body works. Now, one thing I want to mention is that organic molecules can also dissolve in water. So glucose is an example of an organic molecule. So we're going to draw glucose here, and I just want to mention that glucose is a 6-carbon chain that's often found in a ring structure. So this is sort of a simplified version of the glucose molecule. Now, what I want to point out is that I think we could agree that if you drink hot coffee and you place sugar in the coffee, the sugar will dissolve, right? So glucose, it's, I think, believable that glucose would dissolve in water. And I just want to focus on the similarities here so we can see that the OHs that we find surrounding the carbons in the glucose molecule are very, very similar to the OHs that we see in the water molecules. So this is sort of a like dissolves like type of situation. The OHs are similar enough from the glucose molecule to the water that they like to associate with each other. Hydrophilic is the term that we use to describe water loving. So we would say that glucose is hydrophilic because it will associate with and dissolve in water. We also use the word hydrophobic to mean water hating. So when we think of carbons and hydrogens instead of the oxygens and hydrogens, those tend to not like water so much and not be soluble in the water. And so we'll focus a little bit more on this concept when we talk about the different organic compounds. Next, let's talk about inorganic acids and bases. So one thing that's important to note about acids and bases is that there are actually 3 different accepted definitions for acids and bases in chemistry. We're actually going to only address 2 of the 3 definitions, and the definition that we're going to leave out is by far the most liberal, but it just won't be incredibly relevant to our discussion at this point in time. So an acid is a substance that breaks apart in solution to release a hydrogen ion. I really want to just take a moment to talk about hydrogen ion in detail. I know we mentioned it in the previous video. So normally if we consider a hydrogen atom, we know that that hydrogen atom has one proton and one electron. If we were to, for some reason, eject that electron, then that hydrogen atom would no longer have an electron, and now we would essentially call it an ion. So we would have one proton and no electrons, so that leaves us with one positive charge. So we refer to that as H plus, or hydrogen ion. And I just want to also point out that H plus is equivalent to the P plus that we originally discussed as the lonely proton in the hydrogen atom. The reason why I think it's important to remember that H plus is equivalent to just saying a proton is that we think of certain medications such as proton pump inhibitors that can be taken for acid reflux. When we refer to those proton pump inhibitors, what we're essentially saying is that we're limiting the entry of hydrogen ions into the stomach. Proton pump inhibitor is a good reason to remember that proton and hydrogen ion are the same thing. Another way that we can define acid is by referring to it as a hydrogen ion donor. A base is a substance that breaks apart in solution to release a hydroxide ion. So I just want to draw a chemical reaction really quickly here so we can take a look at water, and we can actually break water down into H plus and OH minus. And OH minus is just an oxygen bound to a hydrogen with a negative charge, and that is the hydroxide ion. So a hydroxide ion can combine with a hydrogen ion to make water. The other definition for base is to be a hydrogen ion acceptor. Now, one thing I want to mention, just as a little tidbit, is that technically speaking, whenever we talk about chemical reactions involving hydrogen ions, we will just draw them as H plus. And in all reality, when we think about a couple of different water molecules interacting with each other, what actually is going to happen is one water molecule is going to steal one hydrogen ion from another water molecule, and what we'll have in the end is what we call a hydronium ion, which is actually H3O plus, and then also a hydroxide ion, which would just be the OH minus. So for simplicity, we are going to always represent hydrogen ions by the H plus, but in all reality, it's really H3O plus that we're talking about. Most textbooks will refer to it as H plus. So let's define strong acid. A strong acid dissociates completely in solution. So again, we're oversimplifying it here, but if we were to take hydrochloric acid, for example, HCl, which is stomach acid, and we were to drop it in water, then every single molecule of HCl is going to separate into H plus and Cl minus, and there won't be any molecules of HCl that are united inside of the water. A weak acid, on the other hand, dissociates partially in solution. Carbonic acid is a good example of a weak acid. So if we were to look at the solution, we would see that we have a combination of the H2CO3, the carbonic acid, intact, as well as some bicarbonate ions and some hydrogen ions. So we've seen this reaction before, but carbonic acid dissociates into H plus and HCO3 minus, but it's a reversible reaction. So the HCO3 minus and the H plus can also combine to form carbonic acid. So this reversible reaction is what contributes to the weak acid in this context. Let's talk a little bit more about acids and bases. If we write H plus with brackets around it, that means that we're saying the hydrogen ion concentration. And the hydrogen ion concentration can also be measured by something called pH. And the pH can actually be measured using a pH scale. So let's just draw the pH scale really quickly. So the pH scale basically ranges from acidic to basic, and with neutral in between. So it's a numerical scale. It's also a logarithmic scale, and we're going to talk a little bit about that in a moment. But it's a numerical scale that ranges from approximately 0 to 14. So 0 would be on the acidic end, and 14 would be on the basic end, and 7 is going to be right in the middle. So one thing that I really want to point out, because this is an inverse relationship and it's very, very critical, a high concentration of hydrogen ions is equivalent to a low pH. So as the hydrogen ion concentration increases in a solution, in the body, in a given situation, as the hydrogen ion concentration increases, the pH is going to decrease. The lower the number for the pH, the more acidic something is. So let's talk about a couple of examples. The pH of water is 7.0, so it's exactly neutral. The pH of blood is 7.35 to 7.45. Notice that it's a range. It's a range because we consider something that is between 7.35 and 7.45 to be within normal limits. The body has the ability to fluctuate slightly in between those numbers, but it needs to stay within those numbers in order to be existing in homeostasis. So you notice that the blood is slightly basic in comparison to water. Saliva has a pH of about 6.5, so slightly acidic. Urine has a pH that ranges from 4.5 to 8. And the reason that the pH can range so much is because the kidneys are able to compensate for a rise in hydrogen ion concentration in the blood or a decline in hydrogen ion concentration in the blood, and can eliminate through the urine more or less hydrogen ions in order to accommodate the needs of the body. And then lastly, stomach acid has a pH of about 1 to 2. So you notice that in body fluids, there's a significant range in terms of the values that are associated with different hydrogen ion amounts or pH levels. All right, so I just want to talk for a moment about the logarithmic nature of the pH scale. It's easy to think about the pH scale in terms of whole numbers. Even though we're using decimals to describe body fluids, it's easy to start with whole numbers. So a pH of 6 is 10 times more hydrogen ions than a pH of 7. A pH of 5 is 100 times more H plus than a pH of 7. And a pH of 4 is 1,000 times more than a pH of 7. So I mentioned that not because I want you to memorize it, but because I think it's important to understand that a fluctuation in blood pH from 7.35 to 7.25 would be considered extremely significant and possibly very dangerous. So the body, when it is balancing its pH and managing hydrogen ion concentration, a small change in those numbers is actually considered very significant. A pH that's less than 7.35 in the blood is actually called acidosis, and a pH that is greater than 7.45 is called alkalosis. And as we progress into more physiology, we'll talk about the reasons why acidosis or alkalosis might develop. And the general categories of disease are respiratory and metabolic. So we're going to follow up with that and talk a lot about those types of diseases. Now, one other thing that's very, very important is that as the hydrogen ion concentration increases or the pH decreases in the body does not tolerate acidosis well at all. And actually, it can cause chemical bonds to break, a change in the shape of many complex molecules, and disruptions of cell and tissue functions. So the body really works hard to maintain a normal and appropriate pH. And if that pH falls out of range, the consequences are essentially that the structures of the body can be destroyed. All right, lastly, let's just talk about salts for a quick minute. So a salt is an ionic compound that consists of any cation except for a hydrogen ion and any anion except for hydroxide ion. So if we were to have an H plus or an OH minus as part of the compound, then we would call it an acid or a base. So that's why we can't call it a salt. Now, a buffer is anything that stabilizes pH by removing or replacing hydrogen ions. So what that means is that the buffer is going to bind with an H plus, and that H plus is no longer free floating, and therefore it's no longer contributing to the acidic environment. So Tums is an example of a buffer. So we know that HCl, hydrochloric acid, is stomach acid, and when we combine that with Tums, our resulting products are going to be calcium chloride, which is like a chalky substance, water, and carbon dioxide. So essentially, we've rearranged the reactants and made products such that the H plus is now contained in the water and it's not free floating, so it's not contributing to the acidic environment. A buffer system is responsible for maintaining a normal pH in body fluids. And commonly we're going to refer to the blood, but there will be other examples that might be useful. The carbonic acid bicarbonate buffer system is a prime example, and we can see H plus that's present in the blood, and it combines with the bicarbonate ion to form carbonic acid and eventually is converted to water. So it's just important to note that this hydrogen ion, again, is being removed from solution by combining with other molecules in order to essentially kind of be neutralized. That actually concludes our discussion on inorganic compounds. So I hope you enjoyed the video. Thanks for watching.